I'd like to welcome everyone to the May 26th, the last meeting of the working group in May. I do appreciate you coming today, and I need to remind you that the meeting is, as always, live streamed on YouTube, and edited versions of the meeting are available archivally, so please be aware of that. Today, I'm the only speaker, so I'm introducing myself. I will try to not talk for too long, and we'll see how things go. Tom Hellikar, who's co-lead with me now after Reinhardt's long efforts in founding this group, is unavailable today, but he and I are always eager to hear suggestions, comments, and ideas for improvements. Jim Sluka and Lorenzo are always here also as part of the leadership to take your suggestions, and we appreciate your feedback. We have our usual communication channels. Please do help us use them. In particular, the YouTube channel needs some love, but is also a tremendous resource with more than 120 talks available. And we would like your help in promoting that, distributing it. And if somebody also wanted to help annotate it, that would be even better. That's a big job. We have the tag that Jim Sluka created, MSM Viral. If you use that in your announcements on Twitter and elsewhere, it will help people find the working group. Are there any general announcements to make this week? I want to congratulate Reinhardt and the other group members for the publication in Nature PJ Digital Medicine of the opinion piece, Building Digital Twins of the Human Immune System Towards a Roadmap. You'll see many names that are very familiar here.
Including yours, of course.
Reinhardt did an incredible job leading this project. And I want to just acknowledge that work. If you go to the homepage of the working group, you'll also see a variety of papers were recently published by Gary Ann and his collaborators and other working group members. And I encourage you to please submit papers that are related to this to put up on the working group. Reinhardt, Tav and I had a nice talk with Reed and Grace Peng from NIH last week, and they said that putting up the results of these working group and associated research is something that's very useful. So please do help us. If you need help getting papers up on the working group site, please reach out to Jim Sluka or Lorenzo, and we'll help you get your material promoted appropriately. I should mention that there is a steering committee meeting tomorrow morning at 11 a.m. Tom isn't here today. Tomorrow I may miss part of the steering committee. I apologize for that. But there will be a steering committee meeting. If you feel you should be invited to the steering committee meeting and you do not receive an invitation, please email Jim, Lorenzo, or me, or Tom, and we'll do our best to get you your invitation. Are there any other announcements to make today? Reinhardt, Lorenzo, Gary, anyone? Reed, do you have anything you wanted to say today?
Just to echo what you said, you know, if you do have issues with the wiki, you can also reach out to me as well. I'm willing to help. I think I have permissions now to do lots of uploads. So, I mean, that's one thing I can do to offload some work on you guys. You can direct wiki updates to me where that's helpful. Yep. And then I think, you know, the other thing is we had a meeting with the MSM steering committee, or the IMEG steering committee. When was that? Last Thursday, right around this time. I actually was at this meeting, which is why I missed it. And I think there's a lot of energy into sort of re-energizing, reimagining the broader MSM. I realize I'm not on camera. 1 second. Reimaging, reanalyzing the broader MSM, everything from you know, the mission that was initially set up in 2005 5, something like that, to kind of like what can be done going forward. And I think there's a lot of energy around trying to get people back together in person. And so I know there was a lot of things have been kicked around during COVID a couple of years ago about this group sort of doing the meeting for IMAG, the broader IMAG, and that didn't get off the ground for a number of reasons. No issues from our group here. I think it was on the other end. And I think that, we're looking forward to seeing if we can make something happen in the fall, for a meeting, or something like that. And so I guess the final thing I'd say is, if we have ideas for what that meeting would be, but if you want to throw some feedback our way, I'll put my email in the chat and you can send thoughts, comments as we try to sort of reinvigorate the IMAG. So thanks.
Thank you, Reed. That's great. I know there was a lot of enthusiasm for the idea of contributing to such a meeting, and COVID kiboshed a lot of the ideas. I know John worked a lot on this, and I think if some of those ideas see the light a little later, we'd all be happy at seeing that, and we want to see this be a success. We all appreciate very much the things that you've done, and your involvement and support for this community.
You're here.
No worries. Yeah, I wish we could do more. I think I think the thing is, just real quick, and I'll let you get to the presentation. But I think re-engaging the steering committee that's part of the broader IMAG, a number of professors around, I think that might have been one of the issues where we sort of had that false start. So I think, their enthusiasm to continue and get things going, I think is going to put a little bit of positive momentum behind productively moving forward this time. So keep you posted. I'll communicate, the interest in immune digital twins as a theme, and then try to make that fit with the broader, there's interest from all different government agencies, but definitely highlight that.
Right, well,
This is
John.
I'm really glad to hear that, Reed. That's good news, and I'm more than willing to help in any way I can.
Absolutely, we'll definitely reach out. I think I get nervous with happy talk, without follow up. So I think our focus right now is to continue to meet on our side. We're meeting weekly, and to continue focusing on. I mean, in my opinion, the most important thing is getting folks together. I mean, I don't know about the COVID variants and how that's going to impact things, but I really think that that's really where you're going to get synergy and action. And so I think my biggest interest is trying to figure out how we can do a meeting, maybe a hybrid meeting, right? But something in the near future.
So I think Reinhardt's Florida meeting on immune digital twins, and then the Paris meeting in late 2022, late and mid 2023 will be critical complements the things you're talking about, Reed. And if we can do more meetings that are related to this to keep the momentum going, I think that's critical.
Yeah.
So I think as a community, we need to work together to make this happen and keep it going. And Reinhardt has these great projects that are already on the books. And so now the question is how to keep supporting that and make sure that the momentum is maintained. Any other announcements or comments before you hear me continue to talk? Okay, next time we have Gary on the books. We appreciate that. Lawrence Calzone from the Institut Curie on June 9th. Fiona McFarlane from St. Andrews, of course, worked with Mark Chaplin and has done beautiful work on cell death pathways. We'll be talking on June 23rd. We don't have somebody for June 16th yet. As always, you've heard me say many, many times, volunteer yourselves, volunteer your students, volunteer your postdocs. Feel free to speak a second time. Gary's speaking again. Others can speak again. Suggest people you'd like to hear. They could be journalists, they could be experimentalists, they could be clinicians, they could be modelers, they could be software engineers, they could be standards people, they could be validation or FDA people. Anyone you would like to hear, in this broad community, please do suggest it. And Lorenzo is incredibly good about reaching out and getting people to speak. So please do not hesitate to do that. So we're going to move on to me. And I have to say that I was a little bit, and I'm going to start switching over to my slide deck now, if it's okay. I was a little bit reluctant initially to give a talk, mainly because, as I say, I don't think that you need to hear me. You need to be hearing me that many times. It's probably not the most exciting thing in the world. And also because we had talks given by TJ, Giuliano, Jim Sluka, and others, which I think addressed a lot of the issues. And in fact, we heard a talk last week by a researcher at Emory University who was using CompuCell as a modeling framework. And so today I'm going to be talking not so much about science, but I'm going to be talking, did the screen share work all right before I get started?
Yep, looks good.
Okay, actually I'm going to do the share, the split screen. That's all right. Is that okay for people?
Yeah.
Great. Yep. Okay, so I wanted to talk today about CompuCell. CompuCell is a tool that we have been developing for 20 some years as an infrastructure for multi-scale modeling. And I'm going to be doing a little bit of simple demonstrations and idea with CompuCell. There's going to be a tiny bit of science, but primarily the science was talked about by others. So I'm going to be talking a little bit about the infrastructure today. Many people have worked on this project over the years. If I've left out somebody who should be here, please let me know. Several people in the room are on this list, and several people in the room probably will help me a bit with this talk. TJ has volunteered to put the links into the chat as appropriate. So you see here, TJ. Okay. So the first thing that I wanted to say is that while I'm going to be going a little bit fast in demoing, this is designed so that you can follow along. And if you want to follow along and run CompuCell, then the best way to do it probably is to go to the site nanohub.org. And let me grab the link here and put it in the chat. Nanohub.org. And you can register there. It takes about a minute or two, and you can register there while I'm doing my preamble if you want to try things out. If you're not in the US, it takes a little bit more effort to find the right registration method. If you're within the US, you can register through your university. If you're not in the US, there are some other registration options. It really only takes about a minute. It's free. It doesn't send you a lot of emails afterwards. This is a wonderful open source platform that was funded by the National Science Foundation for the past 20 years that allows people all over the world to use server computers to run various kinds of engineering and science demonstrations, software demonstrations. If you would rather try putting CompuCell on your website, on your desktop, you can do that as well. There is a one-button installer for CompuCell. If you want to try it, it does take a little bit more time, which is why I typically use the NanoHub deployments when we're going to do this. And I'll give you a heads up that if you decide to install locally, both Windows and Macs will give you some security warnings. And so you have to override the protection features to allow the installation, but it is safe. We're not going to be putting a virus on your machine. So if you go to the CompuCell homepage, in the left-hand menu bar, you'll see download sources and binaries. Click there, that will take you to the installers for Windows, Macs, or Linux, most common flavors of Linux. And so I'm going to talk very generally for a few minutes to give people a chance to try these out if they would like. And if people have problems with it, they can reach out to Joel or Jim Sluka in the chat and do that. The slides will also be available later if people don't want to try it right now in real time, but do want to try it on their own. So as always, we are talking about multi-scale biological questions. CompuCell was originally developed primarily to study embryonic development. That morphed into modeling what I would call developmental diseases. The classic one is cancer, homeostasis, toxicology, and infectious diseases, and potentially bioengineering, all of which involve interplay of biophysical mechanisms and molecular biology, gene regulatory networks, signaling and networks. And of course, you've seen this slide many times in many forms. Ultimately, the goal of these kinds of computational strategies is to build reasonably realistic replicates of systems, what we would call in our world virtual tissues, that can be used to predict changes in physiological state. Here at the multicellular level, I don't want to go into repeating things that you've heard many times. One problem always with biology is its heterogeneity and its range of scales. Really, to understand biological systems, we need to understand their context, and that requires going upscale and downscale in a way that makes computation heavy, as people in the room know. What we call virtual tissues typically deal with 2.5 to 3 scales. We'll have a subcellular network that controls the cells. The cells will exist, have spatial structure, and do things. So it's a multicellular organization. They live in an environment which could include extracellular structures, chemical fields in the extracellular space, and potentially that would all be embedded in PBPK pharmacokinetic model of transport in the body if we're going to do things like drug dosing studies. I'm going to go through all of this quickly because people have seen most of this before in other contexts. There are a lot of simulation packages, a growing number of simulation software packages that allow you to do this kind of models. CompuCell3D, as I mentioned, has been around for almost 20 years. The original paper which led to it was published almost 30 years ago. This is the 30th anniversary year of this method, which is a little bit frightening to me. But they're wonderful packages from Dresden, Morpheus. We've heard talks by people who use Paul Macklin's PhysiCell package, Roland Merck's Virtual Leaf. There's lovely work using vertex models by Okuda and others in Kyushu. Kasia Resniak and her colleagues at Moffitt do a lot of more finite element type models. And so there are many, many different approaches. Simune at NIAID is a package developed by researchers at NIAID. So there are many, many possibilities that all have their particular strengths and focus areas. They're not that different from what I'm going to show you, though. And so the things I'm going to show about CompuCell today carry over pretty well to these other packages. CompuCell itself is a virtual simulation environment. We designed it to try to solve the problem of making model specification as easy as possible. I'm not sure that that's actually the problem that really needs to be solved here. There may be other things that are more difficult, but it was designed to have a customized editor for writing models that supported a lot of the things that you need to do when you're building these complex models. And then a visualization tool called a player looks a little bit different on a desktop and in the NanoHub deployment. The goal here was to make model specification compact and where possible to use standards for component model specification. Not all of these models are standardized. That's something that would be a great thing to discuss in the future. Again, it's something that you can try out. CompuCell has support for certain kinds of primitives that you'll encounter in biology, chemical fields and extracellular matrix fields, so contiguous materials, cells and their components, which we call generalized cells, things like cells, nuclei, leading edges, apical and basal surfaces of cells, the mechanical structures like filopodia, junctional adhesion, and so on, which we call links, and then dynamic networks, which is the control and regulation of these systems. This is a relatively flexible package. It's used by a reasonably large number of people. I think there were 22 publications last year using it. A few of those come from our group, but most of them come from groups around the world that use these methods. Here are some of the package papers that have been written in the past year about this. As I mentioned, we've heard a few talks by different groups using this method. So what I'd like to do today is show you what the code what it looks like to run it, a little bit about what the code is, and let's just see what we can do with it. So to begin with, if you want to try this, you go to nanohub.org, tools, CC3D demo, CC3D along demo, and the link is in the chat. And so we will do that. And I have to find the version of it that I have. Here it is. That link will take you to here. And you'll hit Launch Tool. And if everybody does this at once, it may take some time because we're in a shared server. And you should see something like this. And why don't I just give people 1 second to try this out? If you just want to watch, that's fine too. It will pop up a little simulation that we've written of angiogenesis. We are simulating here HUVEC cells, human umbilical vein endothelial cells in culture. These cells are going to secrete a chemoattractant. They're going to chemotax to the chemoattractant. So this is autocrine response to a chemoattractant, which is believed to be the way that endothelial cells in the peripheral vasculature form capillaries. And we have here in the simulation the ability to change parameters as the simulation runs. And the first thing we can do is we can change target length. That changes the aspect ratio of these cells. And the reason that this changes the reason we want to do that is that we know that HUVEC cells actually, as part of their process of forming vascular networks, do elongate. We can also control the strength of the cell's response to the chemical gradient that they're creating. And here I'm going to increase the strength of the chemotactic response. We'll see that the cells will aggregate. And if I let this run, we'll see that they form chords, which are what you see in cell culture when you do this experiment. This experiment I used to do when I had a working wet lab. We did quite a bit of work with HUVEC cells. And so this is an example of a package simulation that allows you to explore some basic biophysical mechanisms and signaling between cells in a compact way. So that's what a typical CompuCell simulation looks like. If you were running it for yourself, you might interact by editing text rather than through a slider. The sliders are really designed for easy user interface. So let me go over some of the features and capabilities of CompuCell. We've seen Player. I want to talk about Twidit, which is our model editor, for a minute. And we've seen CompuCell launch. And I'm now going to switch to a different version of CompuCell, which is our base model here. Let's see if I can get it to be open. Let's see. Oops, that's not going to be good. I have it here. I have too many things open at once. Our base model here. And within CompuCell, did somebody put base model here? You can actually do this within the one we just showed if you're there and you would prefer to work with it. We're going to launch CompuCell3D main tool. This is the bare environment. And what I want to show you how to do is to use our editor. We don't have full GUI-based model specification, but we've created GUIs that allow you to do a lot of the model structure definition. And I want to walk you through very quickly what it looks like to build a very simple model. And the model that we're going to build is going to be one about cell growth and proliferation. And I want to show you what it looks like to build this simulation. I've actually got it built here, but I'm going to rebuild it. To do this, if you're in NanoHub and you're running Player, you have to turn on Twit It. Twit It you can get by hitting this little icon that says Start Twit It up here, or you can do it under the File menu as well. If it's already open, you have to switch tabs. And here we are in Twit It. This is, as I say, a model editor. If you're on desktop, you search and launch Twit It++. And now, and with NanoHub, occasionally you'll have an issue with the frames and the screen size. And if that happens, joggle the bottom right-hand corner and it'll redraw and clean it up. So what I want you to show you is how to build a new model. To build a new model, we can go to CC3D project, hit new CC3D project here, and give the simulation a name. New simulation 2 would work. Doesn't matter. Hit next. The first window that we're going to get will specify the size of the simulation. It's a lattice-based simulation, whether we have flux or no flux boundary conditions, how long the simulation runs, and some other details. We'll accept the defaults. So hit next. We have to tell the simulation the kinds of cells we're going to use. In reality, we might have T cells, B cells, vascular cells, and so on. I'm just going to call them cell 1 and cell 2 for the moment. We add the cell types to our simulation. Not the most thrilling thing to do, but it works. We'll hit next after we've added the cell types. We can create one or more chemical fields. If I were to specify chemical fields, it'll ask me various questions about how those fields work. Today, I'm not going to demo that. I don't have time, although that's in the simulation I already showed for angiogenesis. So we'll skip that. And we come to a window that asks, what are the basic attributes of the cells that I want to have? In these constructive models, nothing exists unless we assert it. So if we don't say the cells have a volume, they don't have a volume. So we have to think about things that we might take for granted otherwise. We'll say that the cells have a contact energy, that is, they stick to each other, that they grow, that they divide, that they have adjustable volume. So I click those 4 boxes, and I hit next. And I'll get a message that says the configuration is complete. I hit finish. And I will have something here called new simulation. If I double click here, I can see the simulation code that this generated. And now I'm going to run that simulation. To run it, I can double click on that name of the simulation. Here I can select open in player. And it's going to give me a message saying simulation still running. I want to stop and restart. And this is actually that simulation running from a previous version that I was testing earlier. I'm going to hit step, a circle, to restart the simulation. I have a redraw problem with the user interface here with the server. And let me see if it's going to allow me. Sometimes it doesn't play nice with it doesn't play nice with Zoom. If I redraw, if I move the corner a little bit, it will redraw. I can use this Windows tile function to lay out the screen. And what I'll see here is an array of cells of type 1 and 2 that have been defined by my model. And I can now hit run, and I will see there's an initial relaxation. The cells will grow. They'll divide when they reach a doubling volume. And if I watched carefully, I would notice that the cell types change. If it's originally cell type 1, it becomes cell type 2 after division and vice versa. I could pull down some menus, for example, to change the colors and the visualization, to save movies, and so on. And that's all it takes to write a simple simulation. It doesn't take very long to do the specification. I want to talk a little bit about what the code looks like. This is just a very quick fly through of some of the ideas you'll see in the code. CompuCell code is divided into two parts. Markup language part, which specifies static properties and is typically designed to be human readable and standards based. And a Python part that's more flexible, but requires a little bit more thinking to build. If we go to the XML part here, we'll see this. We'll see that there's a basic block that tells us things like how we parallelize the code, if we're parallelizing the code, the size of the domain. This is basically the frame that we used at the beginning of the simulation. Then we have our list of cell types that you'll remember creating as well. Again, nothing exists unless we assert it in these simulations. And then we will have to specify what the behaviors of the cell are. Here we specify the adhesion between each kind of cell and the cell in its environment. And finally, we specify our initial condition. Here we drew a square with two kinds of cells in it and defined the initial size of the cells. So that sets up the initial configuration of the ladder. If we switch over to the Python, we'll see boilerplate for Python. Again, we try to hide the Python boilerplate so that you have a minimal need of Python in these simulations. The concept of the Python CompuCell is that you have a start function that's run once at the beginning of the simulation that you use for initialization, a step function that's run each time step of the simulation, and potentially a finish function that is called to save data or otherwise at the end of a simulation. Something that you typically do in a CompuCell simulation here would be for-cell and self.cell list. You're iterating over all cells in the simulation. You're assigning them a target volume and a compressibility. And that's a typical kind of thing that you have to do when you're setting up the initial configuration of your cells. The cells don't inherit automatically things like compressibility or volume. The step function here has a growth steppable. Step functions tend to be called steppable. The growth function goes over all the cells and has them grow at a constant rate. Of course, that's not realistic biologically. In reality, the cell growth rate would depend on the environmental conditions, the nutrient status of the cell cycle, and so on. We can add that kind of complexity later. But again, we have to do this in a very simple way. A little bit more elaborate is what we do if we want cells to be able to divide. Here we have something called a mitosis steppable that knows how to deal with the creation of a new cell when a cell divides into a daughter cell and a parent. We create a list of cells, empty list of cells that might divide. We iterate over our cells. Here we have a toy condition, which is that we're going to divide the cell if its actual volume is above a target. We create a list of the cells we're going to divide. We run through the list of cells that we're going to divide. Here we're dividing them in a random direction. We could do that in other ways. And then once we've divided them, we have to say how the daughter cells inherit from the parent. And here we're saying that each of the two cells that's created has half of the original volume. And clone parent to child means that the daughter cell has the same properties as the parent otherwise. And we're going to do something a little bit of a game. If the child parent cell was of type 1, the daughter cell will be of type 2 and vice versa. So we have a flicker between cell type 1 and cell type 2 on cell division. That's why you see the cell types change when we run the simulation. And that's it to run the simulation. And again, we could come back and look at the output of the simulation. If we had a little bit more time, we could go in and edit each one of these things. And in about an hour, we could build a little avascular tumor simulation where the cells grow, they're subject to contact inhibition, they pull nutrient and oxygen from the environment, they potentially become necrotic or quiescent as well. So this is the basic kind of structure that you use to build these simulations. One thing that TWiDIT has, which I wasn't really going to introduce very much, but I'll just put it here, is it includes a concept of code snippets. That is, most of the things that you would like to do, measure the cell volume, go over all cells in the simulation, plot the values of the parameter in a plot. There are what we call code snippets, that is predefined Python syntax with an explanation that you can paste in to the simulation. So you don't have to remember, everything. Look it up. That's right. There was a question about did you have to handwrite the specification of cell division? The thing that I used was default boilerplate code for cell division. If I wanted to do more meaningful cell division, then I'd have to modify that. But the wizard generates a boilerplate code that has working version of cell division. So that really is a simple explanation of CompuCell and how it works. I can't go into the details today about coupling to network dynamics, but CompuCell has a very easy way of installing networks either in the whole simulation or in each cell individually. You can load, for example, an SBML model from the biomodels database. This is the Tyson cell cycle model. I can run a version of that model inside each cell independently and have it control the cell cycle. This would be much more realistic than having the cell grow at a continuous rate and divide at a fixed doubling volume. And so we can pull from the biomodels database. Here we can pull it directly from the biomodels database into CompuCell and run it inside each cell. When the cells divide, we can have them automatically inherit the state of their chemical networks from their parents, which is a very powerful feature. We could also write those subcellular networks in the antimony model specification standard. That has some advantage compared to SBML, is that SBML is written as an exchange standard. Antimony is a programming specification, which is more human readable. Here, I'm creating notch and delta, and I have Hill functions or Michaelis functions for how those are controlled. Those are coupled between cells. So delta and notch function is signals, juxtacrine signals between cells. Again, I can load the model into each cell and then have the model's pattern. And in fact, we've heard Lorenzo give a talk using CompuCell, taking this simple delta notch model and making it much more realistic in the context of the experiments he's doing. More recently, and this is something of course, Anna is very connected to, we've integrated the MABOS stochastic Boolean solvers into CompuCell. Again, you can run them between cells or within cells. And this is an implementation of their famous delta notch and cell cycle models running inside of CompuCell. And here is the code to do that. Again, you load the model into each cell. You have simple rules for reading and writing its state and for executing the models. The last thing that I wanted to do, I'm already almost out of time, in fact, I probably run over time a little bit, was just to show you one other model that we have This was a simulation that was based on simulation that TJ and Giuliano have presented. Let me see if I can find it here, called Drug Treatment Explorer. And, sorry, I got it here. Find it. Drug Treatment Explorer, here we are. This is an example, maybe not the most meaningful example, of a simulation of a tissue patch. Here we have a tissue patch. We imagine lung epithelial cells. We start out with a single infected cell in the middle. And we're going to ask the question, if we give it a therapeutic, an antiviral therapeutic, at a particular dosing schedule, what happens? And one disadvantage of this particular simulation is that there are a lot of parameters you might want to change. The drug could be something like remdesivir, which blocks RNA replication. It could be a drug that blocks internalization of the virus. Remdesivir is a prodrug, so you have to model the metabolic rates of the conversion of the prodrug into drug, and also the rate at which the drug is eliminated. And so there are a lot of options here. The one, if you're going to do this, that I would recommend playing with is this one here, replication blocker dosing period. You may have to scroll to the bottom depending on the screen that you see. If you get a screen that's really hard to read, go to Windows and hit Tile, and that will relay out the screen. You can then adjust the components. And here I can ask the question, if I give the drug every so often, and we have to know what the units of our time are, what happens? And so let me run this simulation here for a period of 26. Here, 1 microstep corresponds to 1 hour. So this would be about a daily dosing of this drug. And I should see, let me see if I can resize this so I can see better. I can see here, I'll use the tile function to lay things out a little bit better. I will see here viral replication rate, the dose of replication blocker within individual cells, the number of cells that are dying. My cell field is a little bit hard to see here, so let me zoom in. I see a single cell that's been infected and is releasing virus, actually died already, it's yellow. Here I see some cells that have become infected. I can look at the viral concentration as a function of position. I have a model here of cytokines as a single cytokine. It's a simplified model. And oxidation produced by killer T cells and macrophages and CD4, CD8 plus T cells. And I can then begin to explore how this drug treatment, given at different times and different dosing schedules, affects the spread of the virus and the death of cells within my tissue patch. We would like to be able to go further with that. This particular simulation was written some time ago, and it implements this model that we wrote some years ago, plus the drug efficacy. One thing that we did more recently, and Giuliano talked about this, was to ask the question for a prodrug like remdesivir, if you change the uptake rate or the metabolic rate cell by cell, which we know is true, what does that do to the drug efficacy? And in his study, he showed that if you have heterogeneity in drug metabolism, you need a higher dose of the drug to achieve the same level of control of the virus on the same dosing schedule with the same time of initiation of the treatment. And he did a series of studies of that. This actually would be a really easy simulation extension for you to do. And so one of the things that we found, which is perhaps not surprising, is that the few cells that have the least production of the drug to the prodrug to the active form produce the most virus. And therefore, the infection is essentially controlled by the few cells that are most inefficient at producing the drug from the prodrug, or simultaneously the drugs that clear the drug the fastest. And this could be a little homework assignment. You could use Twidit, go into the version of this here on NanoHub, I could go to open this and quit it, and I could change the metabolic rate constants in my models, and that takes about 4 lines of code to do. And then you could actually do the whole study that I outlined here with 4 lines of additional code on this model. Today, I don't have time to do that with you, but I wanted to give you a little bit of a flavor for what one can do. This simulation, of course, is a lot more complicated than the one I wrote down before that we did together. But fundamentally, its components aren't very different. It's a framework. Again, these are designed to be extensible, reusable code. And so if somebody in the room tried to download this code and run it and explore it and extend it, we'd be delighted. It's all open source. So this kind of virtual tissue can work as a variable power microscope. There are a lot of things that one can do with it. It has some disadvantages. It's relatively slow. The number of cells you can simulate is in the thousands, tens of thousands. And there's a lot of complexity to building these models. Something that we've been working on a lot lately is a faster, more elaborate simulation environment. TJ's worked on Mechanica. That's now released. If you want to try it out, that's available. It's a center model like Paul Macklin's PhysiCell, but it's going to be extended pretty soon to include subcellular element models and vertex models. The other thing is that if I look at Player, when we developed Player, the notebook concept wasn't so familiar. Remember this 20-year-old package. These days, people like to run Python in particular inside of Jupyter notebooks. And so we're working on a release of CompuCell that won't use the Player interface. It'll run inside of Jupyter notebooks. That'll make things a little bit easier, especially for educational purposes. If you want to try these things out again, you can go to CompuCell3D.org. You can try it out on NanoHub. We teach an annual summer school. This year it'll be August 1st to 7th. It's going to be virtual. It's free. Send yourself, your students to take it. We'd love to have you. If you want to see what last year's course looked like, make a decision about if you're interested. The 2021 course is available on YouTube. So you're welcome to try that. I'd like to thank NIH and NSF and other agencies for their support over the years. And I need to do my usual disclosures that through my pension fund, somebody bought some Gilead stock. And since I use the word remdesivir, I have to disclose that. And some of the other people involved in these projects have other disclosures related to pharma. So with that, I want to thank you for listening. I realize I ran over a little bit. And this is not our standard scientific talk, but again, enough people had used CompuCell in their scientific research that I thought you might like to see what it looks like to actually build a CompuCell model and run it, not just see the results. And with that, I guess I have to introduce myself and say that discussion is open. Thanks, everybody. Sharon. Is that a question there?
No, that's just the applause emoji.
Lorenzo, you use CompuCell, so you can. Or Chantal, is there a question?
Yes, I was gonna ask, is this all a 2D domain, or do you have also, can you simulate like the thickness?
I'm glad you
tissue?
asked that question. So CompuCell is intrinsically, I don't know how this will work in terms of display here on the fly. CompuCell intrinsically is a 3D simulation. It's a lattice-based method, so you make it 2D by making it 256 by 256 by 1.
Okay.
But I could perfectly well make it 256 by 256 by 256. Of course, it'll run slower when I have all that extra space. But it is intrinsically 3D, and most of its features know automatically whether you're running in 2D mode or 3D mode. These virtual tissue simulations that I showed are what we would call 2.5D, which is two 1D layers on top of each other. And for example, if you think about the surface of an epithelial sheet of cells and then the mucus just above them, that can be relatively conveniently compressed into a two-dimensional layer in the cells, a two-dimensional layer in which the virus and chemical signals and immune cells are moving. So 2.5D simulations are also possible. But 3D simulations are built in. And if you wanted to explore full 3D simulations, in the demos, there are a number of full 3D simulations. That vascular simulation, there's a 3D version of that as well, to do 3D vasculature. Again, it runs a little bit slower. There is rendering in 3D, chemical fields with isosurfaces. Those all exist in 3D built in.
Okay.
Gary.
Thanks, James. I may have missed it, but do you have like a computational requirement limit? Like how big a simulation is it tractable to be run? Can it be run on HPC? Can it be distributed across different processors? Can you do those things?
Okay, good question. So actually, we wrote an MPI version of this 25 years ago, and no one used it. Because these are stochastic simulations, and typically you're doing large parameter sweeps. And so it's more efficient to be embarrassingly parallel and keep each simulation on one box and not worry about the message passing overhead, at least in those days when MPI was slow. CompuCell is intrinsically multi-threaded. And so if you have a very fancy processor with 64 cores, you can tell CompuCell to use 128 threads and it'll run about 60 times faster than it would with a single core. It's actually that's OpenMP. Its OpenMP parallelization is very efficient. And even on NanoHub, which is not designed for high performance computing, I'm allowed to request 4 threads. And so if I went into that early metadata section of the code and said, give me 4 threads, I'd see about a 3-fold speed up in execution. There were in the past some GPU solvers, especially for diffusion, which is naturally very GPU friendly. Those again were not used a lot. And so they've gotten, and GPU programming languages have evolved. And so we're in the process of refreshing all of the GPU support. The graphical rendering, we've had a very, very good master student who has written both OpenMP and is now doing GPU implementations of all of the visualization. Visualization, especially in 3D, the visualization is time consuming. You can run easily thousands of cells in 2D or 3D. Tens of thousands of cells means it's more overnight. And so if I'm going to do a demo here, I want something that runs in real time. If I'm willing to wait overnight for my results, I can run tens of thousands of cells. And if I'm willing to wait a day or two for results, I can run hundreds of thousands.
Per run?
Yes.
For like how long a run? Like a week? I mean, simulated time, like 10 days, 20 days?
Well, so one question is how much time, you mean how much time am I simulating?
Or how many steps? Let's talk about how many iterative cycles.
Well, so here I've gotten to, right, I stopped this, but I'm at 700 Monte Carlo steps. You can see the Monte Carlo steps advancing here. Each one of those steps here is representing a couple of minutes of real time. But this is a fairly small simulation. Right. So it's got how many? It's got 1,000 cells. So if it scales with the number of cells. So if I wanted to run 100,000 cells, it would run 100 times slower.
And can you run multiple grids in the same simulation?
So that is a limitation. This is a fixed lattice method. You can do things that are a little fancy in terms of if you have simulations where there's a lot of empty space, CompuCell allows you to move the grid around you. So if you, for example, were simulating a cell crawling in a substrate, and the cell got to the edge and you didn't want periodic boundary conditions, you can tell it to create new lattice sites ahead of you and to get rid of the ones you're not using. And so you can actually adjust the lattice around your cells. If it depends on, of course, one of the things is this is a general purpose environment. We can't always anticipate exactly how it's going to be used. But there isn't the kind of multi-gridding that one would have if you had a full mesh. And in particular for diffusion fields, you might want to have a higher density of mesh where the diffusion gradients are steep and a lower density elsewhere. And that fundamentally, you're trading memory because you're having memory locations that are being used to provide information you don't need against the simplicity of the code. So that's a reasonable question.
Right. So the specific sort of architecture I'm thinking about is related to the digital twin, the multi-organ digital twin concept, right? Where you could imagine that you would have different grids representing different organ systems or sets of tissues in that stack that you're talking about. They, however, might communicate with each other through, the blood, nerves, or whatever that could be represented with, differential equations that, provide aggregate inputs into those, different environments, et cetera. So one of the challenges that we've had with we've had to hack together in other toolkits, right? Different instances of a toolkit that are linked externally with, some ODE or something like that that's running on the outside of it. So I was curious whether CompuCell, has that potential extensibility because of this interest in multi-tissue things.
So there are two things that I'll say. First, that it's a little bit of a hack. TJ now has re-implemented CompuCell in a way that you could write the components as callable and they can interact. So you could do what you described. If you're a little bit less ambitious, and it's something that I've done occasionally, is I could take my cell lattice, I could build walls in it, and I could define my domains to be different things. And then I could use the fact that I can write complex ODEs that are allowed to interact between domains. And I could say, in this box here, I'm going to say that my cells actually represent volumes of tissue that are a centimeter across.
Right.
In this box, my cells really represent cells. In this box, I represent subcellular components.
Right. But that's part that you have to manually partition your grid to predefine which portions of the grid operate under which. Okay.
I agree with you that would be a wonderful thing to do. And I have to say, if I could get funding, I would. That was something that's been on several grants we've written, which were not successful. I think it would be great to be able to implement something like, for example, what Denise has done with her multi-scale models of the lung. We've talked about it, and I think we know how to do it, but implementing it, that's actually reasonably complicated to get it to work right.
Okay.
TJ might have an opinion on that. I don't mean to put him on the spot. Any other comments or questions? But Gary, I agree with you completely that that's a desirable thing to have.
I have a comment on this latest one, and then a question. But my comment is that I think nobody prevents you to create a simulation which works as a black box. And then you create 10 of these, which represent different modules. And then you put them together in a Python wrapper or something like that handles the different things. And that's actually probably the most powerful way of using it.
Right, but as Gary says, you have to do that by hand.
Yeah.
We don't have a set of predefined methods or classes that do that implementation. And so I agree. I think one of the complexities of doing that real cross-scale simultaneous simulation is that there's so many different ways people might want to do that. That if you provide a lot of that, if you provide that functionality in detail, it may be too restrictive because you haven't anticipated how someone will use it. If you make it too generic, you're reinventing Slurm or something like that. And so you have some compromise, which is to study how people use it and then try to capture that usage in a set of Python classes that would be maximally convenient. Maybe that's something we could talk about separately, because I would love to see that happen.
Kira has a question in the chat about the geometry. Rectangular boundary conditions, are they?
It's a lattice. The lattice is rectangular or parallelepiped. You can make the lattice square lattice or hex lattice. CompuCell supports the idea of hex lattices, which reduces lattice artifacts. And you can define the boundaries to be periodic or no flux. For chemical fields, you can define additional boundary conditions. You could have periodic, no flux, fixed value, or fixed gradient boundary conditions. But yes, so you can do each side independently of each other. So if you want to have, if you imagine a strip of tissue, you could make the top and the bottom periodic and the edges and the other long ends no flux. For the top and the bottom. Anything else?
Ruchir asks, so you can make a tube?
So a tube, conceptually, you can certainly make a tube by creating periodic boundary conditions in one direction and non-periodic boundary conditions in the other. So you imagine that the simulation is wrapped. Yes, that's possible. And that's certainly more efficient. One issue we have with these lattice-based methods is that if you have something like a cyst, which has a lot of fluid in the middle, you're wasting a lot of memory calculating, storing the empty part of the simulation. And so these lattice-free methods like Mechanica or PhysiCell are more efficient because you only model things where they're cells. You don't keep track of things where nothing is happening. Of course, if you have things like chemical diffusion that's happening everywhere, then there's no free lunch. So the diffusion solvers typically wind up being on lattice, even if the cells aren't.
That was actually my question about Mechanica. You mentioned Mechanica. If you can give a little bit more, is an extension of CompuCell, is something completely different, something that is an evolution?
So Mechanica is something, well, TJ should talk about it since it's his project. It was started by Andy Simoji, who worked for me for many years. We wanted to have a simulation package, one that could do big simulations, 1 billion cells, 2 billion cells, rather than thousands to tens of thousands. We wanted something that was off lattice, and we wanted something that could be adjusted in resolution. And so particle-based methods, center models, could leverage all of the work that's been done on molecular dynamics to make really fast molecular dynamics code. And so here, your particles are basically, your cells are basically atoms. Now you can cluster your cells to make molecules and have each cell be essentially a molecule, a clump of atomic particles. That's called the subcellular element method by Newman. The disadvantage is that if you have 1,000 particles per cell, then you have, and you can run 1 billion cells, then you're back to 1,000 cells. And you don't gain that much compared to the lattice method. The other thing you can do is triangulate the lattice with links, and then you could build vertex models. And vertex models are a very powerful way of simulating, especially epithelial folding and epithelial reorganization. And so Mechanica in principle is designed to support a center model, subcellular element model, vertex model. And then if you do the triangulation a different way, you could do full solids modeling, finite element solids modeling. And not all of that's implemented yet, but the goal was to have a consistent interface. The simulation that was written using one method would be usable in all those methods. You can cross-couple those particle-based methods to CompuCell by defining interaction energies. And so again, Mechanica was written so that it could eventually interface with CompuCell. So you could have a particle-based layer on top of the lattice-based methodology that CompuCell supports. Those are all things that will take a little bit of time to happen. They're not going to happen in release 1 of Mechanica. Okay, anything else? We're over our hour for a single speaker, but I'm happy to answer any other questions. I want to say again that there are many packages out there now. Every package has its own strengths and virtues, and so if you find this interesting and CompuCell somehow doesn't quite do what you need, I certainly encourage you to look at packages like Morpheus or Chaste that are different implementations of the same basic core ideas. They may work better. Or Roland Merck's tissue simulation toolkit. All of these implement the same basic computational methodology that CompuCell does, with different user interfaces, different programming methodologies, and so on. So I hope that was, I realize that wasn't as scientific as some of the talks we've had, but I thought it might be nice to see a little bit of code for a change. And I guess if there are no more questions, we can call it a day. I want to thank everybody for coming and look forward to seeing you next week. Gary, look forward to hearing you next week and seeing you in a couple of weeks as well in person. I'm looking forward to that. And I appreciate everybody's questions and comments. And if you use CompuCell and you have suggestions, our manuals need improvement. It crashed. You like something, we need additional features like the ones Gary suggests. Please do send them to us. We have a pull list of requests. We're always behind because we're a small group, but we do work through those pull requests. And when people identify bugs, we work hard. There is a discussion forum. There's a Reddit forum for CompuCell where you can ask questions and sometimes get them answered. We created a LinkedIn page for it. Like all social media, it's not so easy to get people to use it. There's a Facebook page for it. And we're trying to develop this community, both for CompuCell and more generally for this kind of multicellular modeling. And so if people want to talk about those possibilities and issues, we'd be happy to do that. Any final comments or questions? Okay, well then I'll thank you again for showing up, and I will see people either at the steering committee meeting tomorrow or at Gary's talk next week. Look forward to talking to you again.